So we're going to move on. Now this was also another example given to you this morning where we have our semi-permeable membrane here. Now that brought us to our topic for today. Another subtopic for osmosis is called tonicity. Cell tonicity. Now, what is tonicity, students? Now, tonicity talks about how things move in and out of the cell. The ability of an extracellular solution. When I say extracellular solution, I'm talking about this thing is not in the cell, nor is it there naturally, but it can be allowed in and it can be allowed out. Okay? So it can move otherwise or vice versa. So the ability of an extracellular solution to make water move into and out of the cell by osmosis is known as its tonicity. So an extracellular solution is what is making the water move into and out of the cell. Now, how is it happening? It is happening through the process of osmosis. Remember, we said that molecules move in and out, or the movement of molecules is our topic. So now here we are, we're studying tonicity, right? How is it that water molecules move in and out? Who causes it to happen? That is what we're studying. Now, when we talk about cell tonicity or tonicity, we're talking about three main terms. So, when we're talking about tonicity, three terms that are very important that we should look at are hypotonic, isotonic, and hypertonic. They are used to compare the osmolarity of a cell to the osmolarity of the extracellular fluid around it, right? So we are talking about who is higher in concentration, who is moving in and who is moving out, why they are going to move. So that is what we're going to deal with. Now, when we are using these three terms or these terms, we are considering only solutes that cannot cross the membrane. Okay? We are only looking at solutes that cannot pass the membrane. All right? So those are the three words which we are going to use now. I will suggest that you take them down quickly. Who is doing the screenshot tonight? We'll have the screenshot taken after the class. Hold on. So I want you to take the three terms quickly for me in your books because you will have to explain those there now with what I'm going to show you. So take the three terms: hypotonic, isotonic, and hypertonic. Let's move on. So the first tonic we're looking at is hypotonic. Now, if the extracellular fluid has lower osmolarity than the fluid inside the cell. Now, extra means anything that is outside. So if I will use the word extra in front of something, it means to say that it does not necessarily belong there, but it is added there. Understand? So when we're dealing with osmosis, molecules can either be in or out. Okay, so they are extracellular fluids. So extracellular fluid has lower osmolarity than the fluid inside. So it is said to be hypotonic. Hypo means less than to the cell, and the net flow of water will be into the cell. Good. So who is having the lower osmolarity? In other words, don't let these words confuse you. Anybody can give it a try of what that is saying there. So it's basically meaning that inside here is going to have a lower concentration. So we're going to say low. For L is going to represent low concentration. And H is going to represent high concentration. What kind of concentration of what? We are concerned with water particles or water molecules. So if water, for example, let's say you put a plant cell. This is a plant cell, students. If you put a plant cell inside a petri dish of water, so where is it? Do we have high concentration? High concentration is the water is highly concentrated inside of this dish here, and inside of this plant cell there is your, so let's draw our cell wall. There is a lower concentration. So if here has a high concentration of water, then where would water molecules move? Remember, in osmosis, we are focusing on where the water molecules are moving. So, where would water molecules move? If it is highly concentrated inside this petri dish, where would it move? Yes.
To come out.
Where would it move?
Miss, it is there.
It would move into the... Somebody said it to move from the inside to come out?
No, into the cell.
Water molecules is high here, so the first thing we look at, the first thing when we look at the osmosis diagram, we must know the first thing, where is the most dilute solution. Dilute mean it's not filled with concentration, doesn't have a high density. There are less substances inside. So we're looking at where water is the most to where water is not the most or is less. So water is highly concentrated here, and water is going to move from an area of high concentration, which is here, to an area of low concentration, which is into the cell. Remember, I told you that. Now here, because we're having a plant cell, this is what will happen. The parts of the plant cell will look like this. So we have our cell wall, we have our cell membrane. If water is going to move into the plant cell, it's going to fill up the vacuole. It's going to go into the vacuole. The vacuole will get larger, then it will push towards the cell membrane and then the cell wall. So after a period of 5 minutes or so, you are going to notice that your cell will look something like this. It will bloat up, or it will become turgid. It will become bigger. However, because this cell got a cell wall, it's going to protect the cell from bursting. Remember, the membrane is very, very weak. As you keep putting in more water, it's like a balloon. It can explode or burst. But in this case, it's not going to burst so easily because the cell wall is there to protect it. Remember, the cell wall is like a thick, dead band around it that protects it. So it's not going to burst so easily. It's going to protect the membrane. But if water keeps going in, then it can eventually burst. So what we're concentrating on here is that water is moving from here because it is high here and it is low here. And then it is causing the cell to blow up or become bloated. Now, there are important terms that we need to explain when we're dealing with this. I will send you a diagram after the class to show you how do you explain it. But for now, pay attention. So, we have to talk about, when we're explaining, we have to talk about where the first thing. Please take this down in your books. We need to first of all talk about where is it high in concentration. Where is water high in concentration? So let's make a point in the corner here. Where is it high in concentration? Where is it low in concentration? How would the cell look? How would the cell look? Will it blow up? Will it not blow up? And what can happen afterwards, after it keeps blowing up or filled with water? So 4 things we look at. We can look at where is it high, where is it low? How does a cell look and what can happen? So let's just review that quickly. It is high in concentration. I'm going to ask one person to tell me. Let's have Ashiana quickly. Yes, Ashiana, go ahead. Ashiana with us this evening. All right, I'm not hearing anything from her side. Let's have somebody else. Let's have Asif Ramru. Go ahead. Yes, Asif Ramru. Asif, are you with us tonight?
Yes, miss.
Could you please go ahead and say something?
Miss, what are we talking about again?
You gotta be kidding me, right? You have to be really kidding me. I am asking you to explain this diagram here to me and you're asking me what I'm talking about again? So you're going to help me through this diagram here, okay? So pay attention. Now you are going to be my companion for the rest of the class. So this is going to be a lesson for the rest of you, for those who are not paying attention. Good. And I will call on one of you all of a sudden. Surprising call. So tell me, where is it high in concentration, Asif?
Miss, that will be inside of the cell, miss.
Oh, it will be inside of the cell.
Miss, it's not hypotonic, miss.
So it will be inside of the cell. Hypotonic is inside of the cell.
No, sorry, miss.
Where is, listen to me. Don't jump to don't jump to the topic yet. Jump with me. Okay? I am guiding you.
Yes, miss.
So look at the diagram. We having the cell in water. Okay? We're having the cell in water, and there is water. So there's water here. This is H for water. And here, something is happening, and there is your cell. So tell me, where is high in concentration? Here. Where is high in concentration on this diagram? Where is water high in concentration?
Miss, outside of the cell?
Inside of the Petri dish. Okay?
Yeah, inside of Petri.
Good. And where is water moving into? Or where is it going
The
to?
cell.
Into the cell. So it is low in concentration. Good. Now tell me, what will happen to this cell? How will it look? Let's say after 5 or 10 minutes, you left it in the water. And this is a plant cell, okay?
Yes, miss.
So tell me how it will look. Look at the diagram with the green now.
Miss, it would swell.
Uh-huh.
It would float up.
Look at the parts. Take a deeper look now. Look at the parts in the cell. So there is our vacuole. There is our cytoplasm. There is our cell membrane. There is our cell wall. Okay? So tell me what is happening to these parts here.
Miss, water would enter the cell.
Through the
And through the vacuoles.
So water just jump into the cell through the vacuole. Are you embarrassed in my teaching, Asif? Now this is one of the problems we're having with online teaching. You are there, but you are not there. You need to be there on the screen. Do you understand that? Every one of you. You are there on your computer or your phone. You need to focus what is happening on the screen there. Because we're putting in time for this. So please pay attention. So I'm not going to give Asif an easy go. He's going to get it right. So Asif is going to tell me the correct thing. Where does water pass through to get into the cell? Yes, Asif. Which part does it pass through?
The membrane.
The membrane. Good. You got it. So it passed through the membrane. So if it passed through the membrane, then it's going to go to which part of the cell next?
That would be the
Cell wall.
cell wall.
Oh, you cell wall there after the membrane.
No, wait.
Which part of the cell is the cell wall, Asif?
This is the outer part, miss.
So if you pass through, so you're not going to pass through the cell wall first to get into the cell, and then the cell membrane.
Yeah.
So we now already passed the cell wall and passed the cell membrane. Why are we going outwards? Why are we going outwards? So let me just sketch another diagram here for you so you can get it proper. So here is our diagram. This is label the parts here of the plant cell for me. What is this part?
Miss, I can't see nothing. Oh. It will have the cell wall.
Cell wall. So water is going to pass here first. Now this wall is not semi-permeable, it's fully permeable, meaning that things will just pass through it. Okay? Good. Then what is this part here?
The membrane.
Cell membrane. Are you not sure what are the parts of the cells? Why are you sounding so unsure? Why are you sounding so unsure?
I don't know.
Well, tonight, everybody, your homework is to go back and review the cell. Because you can't be unsure. Every topic leads to the other topic. Okay? So, we have our cell wall, then we have our cell membrane, and then we have the next part. What's this part?
The cytoplasm.
So, you see the pathway which water got to go in? It got to cross the wall, then the membrane, then the cytoplasm. What did you tell me happened? You told me it jumped into the vacuole. Do you see how silly that sound? Do you see how silly that sound? All right. Wonderful. What is this part now? Sorry? So here we have our, what is this part called now, Asif?
This where?
Here, right here, this part here.
The vacuum.
So, there are other parts, but we're focusing on the main parts of the cell. So we're having, let's go back from how water would enter the cell. So first it goes to the, what's the first part?
Cell wall.
Good. Then the,
Cell membrane.
then the,
Cytoplasm.
then the.
Vacuum.
Wonderful. Everybody give Asif a clap. Good. Good. So Asif is my companion for tonight, and yes, I will choose another one very soon. So be very careful and pay attention on the screen. So Asif.
Let's see what's outside. What's the hair?
Okay, good. So here we are with the vacuole. The cell is filled with water. What will happen now, Asif? Water is in the cell. And we understand why water entered the cell, because it is high from the outside and then it is low in the inside. So it keeps entering the cell here. So what will happen eventually? Tell me.
Miss, it will fill up with water, water molecules, and it would reach a stage where it will start to burst, but the cell wall would stop it, or stop it by a small percentage from bursting.
Very good. Wonderful. Very good. So see what happens when you pay attention. So it will be having a lot of water, and then obviously it's going to get large, as you can see from this one compared to that one here. It gets very large, it becomes very bloated, and then it will reach a stage where it can burst, but it will take a longer time to burst because the cell wall is protecting it. Okay, students? So what do we call a situation of such where water enters the cell and cause it to be bloated? We call this situation hypotonic. We said that the cell content is hypotonic. Everybody get that there? So think about somebody telling you, yo, you look very hypo. What does that mean? It means to say you look fluffy. You look very fresh, very bloated. Okay? So hypo means that you would have swell up, or you would have become very turgid. So words that we can use for this to describe this cell is turgidity. It is turgidity. Okay? The cell becomes turgid because it's a hypotonic cell content. Water enters it. As you can see from the beginning, we say that the extracellular fluid has a lower osmolarity than the fluid inside of the cell. Alright? It means to say whatever is outside is dilute than what is inside. Good. So, turgidity is the word we want to use. Rigid is also a word I want to use. Or bloated is also another word. So these are three terms we can explain or describe the cell as. Or in other words, we can say that the cell bulges, the vacuole gets large, the protoplasm is pushed close to the cell wall, which pushes back the wall with pressure and stop the further water intake. When it stops the further water intake, because every cell has a maximum level at which it can reach, and then the cell becomes turgid. If it's a plant cell, it will not burst right away. It takes a longer time to burst because the cell wall protects it. And if it's an animal cell, then it will burst. Now let's take a look at the animal cell quickly. So I'm going to call on one person to explain hypotonic in a plant cell for me before I go on to hypotonic in an animal cell. Who is that lucky person tonight? Yes. Go ahead. One person. Alright, let's have my dear companion Asif again. Yes, Asif, go ahead. Tell us.
Miss, about hypotonic?
Tell us what's happening here.
Miss, where are you pointing to?
I am pointing on the screen where everything is. So water is in the Petri dish with a piece of plant cells. So start explaining to me what is happening there. Because nobody is not volunteering and you're my companion. So you have to explain.
Okay, so a plant cell is in a Petri dish. So the water outside of the cell is more concentrated, and the solution that is inside of the plant cell is lower in concentration. So
Stop there,
water
Asif.
will
Don't
move-
say more concentration. Don't say more concentrated. Right? Use the word high in concentration. Okay? Concentration and
Yeah.
concentrated is two different things. Alright?
Yeah, miss.
Good. So go back again.
Yes, so outside of the plant cell is high in concentration, and inside of the plant cell is lower in concentration.
Very good.
So hypotonic, is where water molecules move from a region of high concentration to a region of low concentration. So water is moving from outside of the Petri dish to inside of the plant cell.
Is it
It has
outside
to
of the
pass-
Petri dish? Stop. Is it outside of the Petri dish or inside of the Petri dish? Now we want to use the word outside of the cell to inside of the cell, right?
Yeah.
Water is high in concentration outside here. See that there? Outside here.
Yeah.
And it's moving into the cell. So when the water moves into the cell, then it becomes a hypotonic situation, not outside of this Petri dish. So go again for me.
The water is inside of the Petri dish is high in concentration, and inside of the plant cell is lower in concentration.
Very good.
So when water moves from the Petri dish or outside of the plant cell to inside of the plant cell, it will be known as hypotonic. It has to pass through the cell wall, then through the cell membrane, then through the cytoplasm, and then it goes through the vacuole. And when water enters the plant cell, it starts to get blown up. But if the cell wall would stop it for a period of time from bursting.
What do we call this whole situation? How do we describe this cell now? What do we call this cell?
This cell is torsion.
Or
Yes. Hypotonic.
Or
Or
it what? It bulges. Okay? Good.
yeah,
Wonderful. Very good, Asif. Everybody, tell Asif something nice, quickly.
Beautiful, my brother. You got it.
Asif, who do you think should be my next companion? Who do you think should be my next companion?
Jonathan.
Very good. Jonathan is should be. So we are talking, this is hypotonic in a plant cell. Now anytime you have celery, you have cucumber, anything that is plant related, this is what will happen. Somebody might give you a question and say, here what happened. I have a piece of potato and I'm going to make French fry and I put it in a container with water. What will happen after 5 minutes? Now, please don't go and overthink what happened to the potato. Miss never taught me what will happen to the potato or what will happen to the carrot or what will happen to the cucumber or the celery. But you have to know the fact that they are all plant material. So this is what will be happening. Is that clear, students? So anytime you're ready to fry your French fry and whatever, please know if you put it in water, water molecules will enter into the plant cell and it will look fresh. And this is the reason why the market vendors will throw water onto their vegetables to keep it fresh. Do you understand that now? Good. Now we're moving on to what happens to an animal cell. Ready, Jonathan?
Yes, miss.
Wonderful.